Next, we're going to look at non-Mendelian genetics, and these ones don't follow the same rules that we've been practicing with. So with non-Mendelian traits, you can have more than one dominant allele, which means that you can have more than one capital letter or big letter. So the first one we're going to look at is called incomplete dominance, and with incomplete dominance, you're going to have two dominant traits. And when you cross them, you get a third phenotype, and the third phenotype, they're actually going to blend together. So it says in roses, the allele for red flower crosses with the allele for white flower to produce pink flowers. So I'm going to make my key. I'm going to use big R for red, since it's a dominant trait. I'm going to use big W for white, since it's also a dominant trait. And then when I would cross those, I would have RW, which would be my third phenotype, which is pink. So I'm going to be crossing a pink and a white flower. So pink is RW and white is WW. So I'm going to take the first parent's alleles, I'm going to put them up here at the top, so RW, and then I'm going to take the second parent and I'm going to put along the side, WW. I want to keep R first, so I don't think I have another genotype when I really don't. So first box, I'm going to have RW, then WW, RW, and WW. So my genotypic ratios, again, I'm just going to list what I have. So I have 2 RW and 2 WW, or I can say 50% of each of these. And then my phenotype is going to be what color it is. So I'm going to say 2 pink and 2 white. And again, this is where having your key is really handy because you can just look back up here. So again, it would be 50% pink, 50% white. Next up, we have codominance. And again, in codominance, we have 2 dominant traits. And when you cross them, again, you get a third phenotype, but in this case, the third phenotype is going to show both of them at the same time. So think like spots or stripes. So instead of blending, you're going to see both at the same time. So in flowers called petunias, the allele for red flower crosses with the allele for white flower to produce striped flowers. So I'm going to go ahead and make my key. So I'm just going to use big R for red again, since it's a dominant trait. I'm going to use big W for white. And then this time, RW, instead of being pink, will be striped. So it's got some red and some white. So they're not blending together. And we are crossing 2 striped flowers. So the genotype for both parents is going to be RW. Okay, so I'm going to go ahead and put those on my squares over here. And again, I just fill it in like a grid. So big R. Since I used R first here, I'm going to keep that consistent. So I have RW, RW, and WW. So my genotypic ratio, I'm just going to list what I have. So I have 1 big R, 2 big R, big W, and 1 WW. Or I can say 25%, 50%, and 25%. And then my phenotypes, I'm going to say what colors I'm going to see. So I'm going to have 1 red, 2 striped, and 1 white. Again, or 25%, 50%, or 25%. Underneath that, we've got blood type, and the reason that we're going to work out blood type next is because this is an example of codominance. So with blood type, we actually have 2 dominant alleles, A and B, and then we have O, which is recessive. So dominant, dominant. If you have blood type AB, it's a codominant, so like in this one, which means you produce A antigens and B antigens, so you're going to show both at the same time. And then O is recessive. So there's 2 ways to have a genotype for blood type A. We can have homozygous blood type A, which is AA, or we can have heterozygous, which would be AO. So there's no little a. Instead of a little a, we're going to use the letter O since that's our recessive allele. B works the same way. We can have homozygous B, which would be BB, or we can have heterozygous B, which would be BO. Again, no little b, we're going to use O since that's our recessive allele. Codominant is just AB. So there's only one genotype for that, and then there's only one genotype for type O blood since it's recessive, which would be OO. So it says we're crossing a man with heterozygous type A. Again, hetero means different, and type A, so that's going to be AO. And I'm not going to write our key because it's right here. With someone who is heterozygous type B. Hetero means different, and type B, so it'd be BO. So I'm going to take the first parent, put it here, take the second parent's alleles, put them here. Now, when I fill in my boxes, it's just kind of common practice to put A first. So I'm going to put A and then B. My second box would be BO. Since B is the dominant allele, I usually put that first. Same thing with this box. I'm going to put A first since it's our dominant allele. And then the last box would be OO. So my genotypic ratios, again, I just list what I have. So I have 1 AB, I have 1 BO, I have 1 AO, and 1 OO, or 25%, 25%, 25%, 25%. And then phenotypic ratios, I'm going to list what blood types they are. So I would say 1 blood type AB, I would say 1 blood type B, since this is just blood type B. This would be type A, so 1 type A, and 1 type O. Again, or 25%, 25%, 25%, 25%. Okay, last one is going to be sex-linked traits. So these are attached to the sex chromosomes. Which are X and Y. Most of the time you're going to see them attached to the X chromosome. So when we think about our chromosomes, if you have 2 X chromosomes, that is going to represent a female. And if you have an X and a Y, that would represent a male. So these alleles will actually be attached to these X chromosomes here that we're going to look at. So that means that females get 2 alleles, where males only get 1 allele because they only have 1 X chromosome. In humans, hemophilia is a recessive sex-linked trait. So hemophilia is recessive. So I'm going to use big H and little h. Since it's recessive, hemophilia is going to be our little h, which means that big H is going to be normal, okay, or not hemophilia. So we are going to be crossing a male with hemophilia. So since it's a male, it's going to be X and Y. And since he has hemophilia, he has a little h. So what I do is I attach that to that X chromosome. So think of it like a superscript. And then a carrier, female. Now, when we have these types of traits, a carrier means that they are heterozygous. So they have 1 big allele and 1 little allele. So they would have 1 big H and 1 little h. Since they have a big H, they're normal. They don't have hemophilia, but we call them a carrier because they carry the little trait. So since it's a female, XX, and since we said she's a carrier or heterozygous, she would have 1 big H and 1 little h. Now, when I split up these parent alleles, think of this as 1 allele. The X and the H don't get separated from each other. So I would have X, little h, and Y. Again, these are only carried on the X chromosome, so that's why there's nothing attached to this Y here. And I'm going to take the female's alleles and put them down here. So I have X, big H, X, little h. So when I fill them in, I try to keep my big allele first just to keep it consistent. So I have X, big H, X, little h. I have X, big H, and then Y, X little h, and X little h, Y. So my genotypic ratios, just list what I have. So I have 1 X big H, X little h. I have 1 X big H, Y. I have 1 X little h, and 1 X little h, Y. And then, or 25%, 25%, 25%, 25%. Phenotypic ratios, I have to list males and females. So I'm going to work through this one by one. So this, again, would be a carrier, but since she has a big H, she's going to be normal. So you could either say one normal female or one carrier female. I'm going to go ahead and put carrier because that's what they had in the problem. But normal female would be the same thing. Let's just go ahead and write that under. This female offspring would have hemophilia because she has 2 little h's. So 1 female with hemophilia. This male would be normal because he's got a big H. And then this male would have hemophilia because he has a little h. So 1 male with hemophilia. Now, when you have problems that are related to sex-linked traits, you have to be careful if they ask you about the male offspring or the female offspring or the total offspring. So if you were to get a question asking what percent of the female offspring would be expected to be normal, then we're only looking at these two. So 1 out of 2 would be 50%. If you were asked what percent of the total offspring would be normal, you would look at all 4 of them. Again, in this case, it would be 50%. Or you could be asked about the males. What percent of the males would have hemophilia? So 1 out of 2, again, would be 50%. So you have to be really careful what they're asking you for in these types of problems.